Hello everyone, good morning, good evening, good afternoon. On this webinar organized by the organizers of the International Conference on Poultry Intestinal Health. We have organized a webinar today with as topic the applications and use of omics technologies and how these technologies can be applied to promote poultry health and productivity. So basically, what we have been doing is launching a question on the website asking people from the poultry industry what is the main topic that they want to listen to, so to which topic they still need much more information, and then this topic came out of it. So this is in fact as some kind of a replacement of the International Conference on Poultry Intestinal Health that was normally supposed to take place actually in this period in Cartagena in Colombia, and due to Corona, we postponed it for 1 year. So still, you're of course welcome next year to join that event as well. So today we have been selecting this program on the omics technologies and have 3 very nice speakers that are very well known worldwide. 3 different speakers from different continents, and I can tell you that for today we had close to 600 registered people that can also download the videos, of course, online. And currently, I think we are with over 100 of persons being logged in. So that is definitely quite nice. If you need much more information on the actual conference that we will have next year, also the full program is also available. So the program with the topics, the speakers, the invited speakers. So also there we already had some abstracts being submitted, so you can also do that if needed, of course. So what I first also want to do is to thank our sponsors for the Cartagena event, but also the sponsor for this webinar, of course. So without them, we couldn't really do this kind of webinars and this kind of conferences. So we thank them for their grateful support. Okay, so I immediately go over now to the conference, to the webinar and the speakers of this webinar. So we have 3 different speakers for today. Ivan Rychlik from the Veterinary Research Institute, Dana Stanley from Central Queensland University, and also Rick Ducatal from Ghent University in Belgium. And basically what the first 2 speakers will try to do is give you some information on the scientific background of the methodologies, and Richard will then also give some information on how these kind of technologies can be applied in poultry health. And yeah, really from the veterinary perspective, he will give you this kind of overview. If you have questions, you can put these questions in the Q&A on the Zoom, so that's very important to mention, and we will select questions and ask to the speakers. So we aim for 20-minute presentations, 3 different ones, and then also followed by question sessions, 15 minutes each, so that we have quite some time for interactions, which is of course very important in this kind of webinars. So first speaker is someone I know very well for a very long time, because we have been working together starting 20 years ago on Salmonella. So Ivan Rychlik is head of the Salmonella group in the Veterinary Research Institute in Brno in Czechia. And he has quite a lot of different interests, all or most related to poultry. He has been working quite a lot on Salmonella before, that's what I just mentioned, also on Campylobacter, but since some years he also has done quite a lot of work on evaluating the role of the intestinal microbiota in health and disease, and also in relation to preventing Salmonella colonization. So without further ado, I want to give the floor to Ivan, who can start uploading his presentation now. Thank you very much.
Hello to everybody. I will now switch on my presentation and also share my screen with you. Well, so I hope that you now can see my presentation, and I will now first continue with my presentation anyway. So first of all, I would like to thank Philip van Immerser for organizing this meeting, which he calls as a virtual event, but I don't think that it's a virtual, it's a real event. And of course, I also want to thank for his activity and the fact that he invited me and thus gave me the opportunity to share with you our experience with DNA sequencing and its application in gut health research. Real milestone in this area can be dated back somewhere to 2008, when company Roche introduced a new platform for nucleic acid sequencing, FLX sequencer, and soon after another company, Illumina, followed this example and introduced their platforms and their instruments. And for both these companies and their products, it is characteristic that within 24 hours they can produce millions of reads, millions of sequences from the DNA which is subjected to the sequencing. Initially, these products were definitely designed for genome sequencing, for sequencing of human genomes, of different individuals, animal genomes, plant genomes, bacterial genomes, simply pure DNA from a given species and definition and characterization of its genome. But sooner or later, somebody realized that the potential of these instruments is much broader, much wider, and that these instruments can be used for sequencing, even for sequencing of mixed DNA samples, which do not consist of a single DNA from a single organism, but which may consist of DNA originating from different microorganisms or macroorganisms. And these, in that case, DNA is purified from an environmental or biological sample containing different entities, biological. Then the DNA is fragmented, and each of the fragments is then, or nearly each of the fragments, 15 million of fragments are sequenced. When you do experiment like this, you resign for genome assembly. You do not go towards assembly of full genomes, but then you submit all individual reads, all individual sequences into appropriate database and get back information that this blue fragment originates from E. coli because of its sequence, that this black fragment, for example, originates from Staphylococcus, or the green one originates from Citrobacter, and the red one originates from Bacteroides. And since you received information for 15, for example, 15 millions of different fragments, different sequences, you can then focus on the number of fragments which are associated with blue bacterium, black one, green, or red one, and you can then reconstitute back the composition of a microbial population, which is pretty fantastic, pretty nice, because you do not need to culture. You do not need to know anything about the population in advance. You only purify DNA, sequence, and get back the information on the composition. The situation can be even easier when you know about some genes, or about at least one gene, here highlighted by yellow dot in each chromosome of each bacteria, forming that kind of hypothetical population. So if you know about the existence of a gene which is common to nearly all, or ideally all, bacteria, there are genes like this, and one of them, very popular for phylogenetic studies, is gene encoding 16S rRNA. This gene is indeed present in genomes and chromosomes of all different bacteria, and this gene is also highly conserved. So its sequence is very similar even between distantly related bacteria like E. coli and and Bifidobacterium. Now, this gene is quite conserved. However, this doesn't mean that it is conserved and identical in its whole sequence among all bacteria. There are parts of the genes here highlighted with blue color which are highly conserved because of the function of this gene, function of 16S rRNA in proteosynthesis. But these highly conserved sequences and parts of the genes are interrupted with regions which are rather variable. There are 9 variable regions within 16 rRNA gene, and these can be used for the differentiation of bacteria forming complex and total microbial community of your sample. Now you then use the presence of conserved regions for design of primers which may anneal with that gene present in chromosomes of different bacterial species. You then subject this to PCR, simple and common PCR, amplify everything, every gene from each bacteria forming the population, and then this mixed PCR product you subject to the DNA sequencing using Illumina platforms, for example. And you get back the same sequences, up to 15 million or even more, and you read the sequences, submit these into appropriate databases, and get back the information whether the fragment originated from E. coli, Bacteroides, Clostridium, and so on and so on. And you can then again reconstitute composition of microbial populations within your samples. These tools are ideal for characterization of communities which are complex in their composition. And one of such examples of such communities is gut microbiota. And this is exactly where we apply these techniques. And in my next few slides, I will share with you our experience, what can be read, what can be done, what can be deduced from when using DNA sequencing for characterization of chicken gut microbiota, chicken cecal microbiota in particular. In one of our very first experiments, we followed development of chicken cecal microbiota in egg layers over their whole egg production, whole life. So here is age of the flock, age of the chickens, and age of the hens in weeks. So we monitored the development of cecal microbiota for 1 year in this flock, when the flock egg production decreased here and the flock was slaughtered. We visited the farm, always collected free chickens and later free egg-producing hens, sacrificed them and collected cecal contents and determined composition of microbiota in cecal contents, in cecal digesta. And we observed the following. Within the first week of life, here number 16, blue color, E. coli was the most characteristic bacterial species present in the cecum of chickens. But the 1 week after, starting from week 2 of life, E. coli became a minority, and new types, new species, new types of bacteria appeared. In particular, these belong to family Lactospiraceae and Ruminococcaceae, both of them belonging to gram-positive bacteria of phylum Firmicutes. When the flock and chickens were approximately 1 month old, 3 weeks old, for the first time in that case, we recorded presence of another group of bacteria, here highlighted in different shades of magenta color. And these bacteria belong to phylum Bacteroidetes, and their abundance increased and reached maximal and then constant abundance exactly at the time point when the chickens reached sexual maturity and when the egg production was starting. The ratio of Bacteroidetes, gram-negative Bacteroidetes, to gram-positive Firmicutes then remained more or less identical and equal 50 and 50% throughout the rest of the life and egg production. For a certain period of time, we considered this as some kind of dogma, development, age-dependent development of chicken gut microbiota and so on. Well, this is a total nonsense. It's a mistaken conclusion. It's a true, correct, but biologically mistaken. Why I am saying so? The reason is on my next slide, and the answer is in another experiment from our lab. And in this experiment, we raised chicks hatched in hatcheries as we are used to, and many of us, nearly all of us are used to, so as they are. And in another group, we placed these newly hatched chicks in a contact with the adult hen. This hen had nothing in common with these chicks. This was egg-producing hen. But in this way, we anyway mimicked and modeled the situation as if these chicks would hatch in a nest. Something for what chicks have evolved during the millions of years of evolution. And when these chicks were 6 or 12 days old, we sacrificed them and looked at the composition of their cecal microbiota. As on my previous slide, chicks which were raised without any adult hen, which would act as a source of healthy microbiota characteristic for Gallus. Microbiota of these chicks during the first or second week of life was formed mainly by gram-positive firmicutes. However, the composition of gut microbiota, cecal microbiota in the chicks which were raised in contact with adult hen was completely different. It was nearly identical, or it was identical, the same as it was as we recorded in a cecal digester of this donor hen. So now you can immediately conclude that these bacteria, gram-positive firmicutes, appearing as among the first in the cecal digester and among cecal microbiota, forming cecal microbiota in common commercial chicks during the first days of life, these bacteria are of non-chicken origin. Those are environmental bacteria species which are capable of replication and multiplication in a chicken intestinal tract as well. On the other hand, and these are bacteria which are highly dependent on the presence of adult hen. These bacteria must be underrepresented in common environment of our animal houses, farms, barns, everything. And if you start to think of probiotics suitable for administration to newly hatched chicks during the first days of life, you should probably consider these or bacterial species belonging to Bacteroidetes rather than those belonging to gram-positive firmicutes, because their environmental sources are rich enough. Here is some problem. These bacteria do not survive in the environment that much, and their transfer is mostly dependent on a vertical transmission from donor hen to the chicks. We then confirmed this or further expanded our knowledge and understanding in another experiment. As we are really interested in chicken gut microbiota and bacterial species which colonize chicken gut, we, in a parallel to sequencing, we started with anaerobe culture. So we cultured and obtained hundreds of clones and different bacterial species in pure cultures, all originating from chicken cecum or chicken intestinal tract, in small intestine as well. And when we had them in a pure culture, we characterized them by whole genome sequencing, and then having the pure cultures, we inoculated with hundreds of pure cultures, one-day-old chicks orally, and 1 week later, when these chicks were 7 days old, we sacrificed them and checked for the presence of the strain which was inoculated by us on a day 1. And we found out that there are bacterial species which can efficiently colonize chicken intestine, chicken cecum, and those are shown here in this slide. The most proficient Alistipes senegalensis formed in our experiment, in this one particular experiment, when we tested this strain, it formed nearly 80% of cecal microbiota in 1 week old chicks when we administered these bacterium on a day 1. But there were also the others which were quite efficient colonizers. If you are familiar with gut microbiota of chickens, humans, or even pigs, you will understand what these bacterial species are, what these bacteria are. If you are not, some of the names may be rather confusing or unknown to you, but what I can tell you that here in this figure, you will find nearly only and nearly exclusively bacteria belonging to the magenta color here. Only these bacteria from phylum Bacteroidetes are efficient in their colonization when orally administered in a single dose to newly hatched chicks. And all gram-positives, which we tested as well, and these are not in my figure in here because these would be somewhere here and more to the right. These did not colonize chicken intestinal tract at all, despite the fact that we provided them in a huge number, high concentration, 0.1 ml orally under the control. So firmicutes, it is very difficult with these to achieve successful colonization, and those bacterial species which are transferred from hens to offsprings to chicks, these are also very efficient in their colonization and successfully and efficiently colonize chicken cecum after a single administration. DNA sequencing can be used for alternative studies, and I have 2 more slides, and this will be all from my point for today. Next slide is another experiment from us. Well, it's not an experiment, it's a comparative study. No experimental interventions in this experiment or in this report and study. In the Czech Republic, private people, private owners, they keep sometimes, not all of us here in the Czech Republic, but some of us in the village areas, they keep small flocks of egg layers of hens for egg production, but for their own consumption. So it's not a business, it's not for market, it's for their own, in their backyards of their houses, they keep hens. And we use this opportunity, we use this fact and ask a couple of owners of these hens to provide us samples, cecal samples, so to provide us hens when their egg production dropped. And we compared by sequencing. We compared composition of cecal microbiota in backyard chickens to the chickens and hens in egg production, those from commercial, industrial, raised and kept under commercial conditions. Initially, the initial hypothesis was that we were interested in bacteria which will be specific, which will be present in microbiota of backyard, outdoor hens, and which would be absent in cecal microbiota of commercial hens. And we were thinking that this would be nice probiotics, well, absence of sources here, therefore suitable for obtaining in pure culture and administration for improvement of productivity and so on and so on. Well, our results were quite surprising and opposite to our expectation, for example. So whenever we sequenced microbiota from backyard, always blue color here in my slides, we observed lower diversity of microbiota in comparison to the cecal microbiota of commercial hens. We do not have any definitive and final explanation for this observation, but what we believe the explanation is, after all, food or richness of the feed available to these hens is much limited in comparison to the hens kept under commercial conditions. Feed of these hens is supplemented with minerals, growth factors, vitamins, everything here. And these extra supplements not only supports the growth of these hens and egg production, but also supports growth of broader spectrum of different bacteria which may colonize the intestinal tract. So diversity was, to our surprise, higher in cecal microbiota of commercial hens in comparison to those kept in backyard and outdoor conditions. But the composition was different, as shown here in this PKA graph and figure. And characteristic changes were outdoor backyard microbiota in backyard chickens was enriched for Bacteroidetes gram-negative, and also for different proteobacteria. But proteobacteria are different from E. coli, other bacterial species, not E. coli. And this enrichment for these bacteria from these two phyla occurred and happened on expense of gram-positive firmicutes. These were more common in a microbiota of chickens or hens from commercial production than those from backyard. When we then compared genera, which form more than 0.1% of total microbiota in either commercial hens or the backyard hens, and look at their enrichment, higher or lower abundance in either type of the flocks, we were really, as I already said, we were initially interested in those 10 different genera which were more abundant in microbiota of backyard outdoor chicks. And when we then thought about the outputs, final conclusions of this study, after all, we were the most interested in these, because those are true genera, very characteristic for chickens, which colonize chicken ceca, chicken intestinal tract, regardless of genetic line, regardless of environmental conditions, and regardless of the feed composition. So we now focus on these genera and species within these genera and consider these as a potential and the most promising future probiotics. Now, in my introductory slides, I mentioned that you can use DNA sequencing for nearly any, or not nearly, for characterization of any microbial population. So it needn't be cecal or gut microbiota only. And we a little bit expand our interest at the moment, and we started to be interested in litter microbiota, litter of chickens. We have performed a single experiment, single monitoring study right now, or up till now, so it's in its very beginnings. But in this experiment, we followed development of litter microbiota in a broiler parent flock. This means that we monitored the development of litter microbiota for the first 15 weeks of life of the flock, and we compared composition of litter microbiota with cecal microbiota of 3-week-old or 15-week-old chickens from this flock. Now you can see in my figure that there are some developmental patterns in the litter microbiota, but my main message at the moment is that litter microbiota is completely different from cecal microbiota, or pretty different. Well, of course, you can find cecal bacteria in litter microbiota, but once the litter microbiota established somewhere around week 4, week 3, week 4 of flock's life, the microbiota in the litter is different. It is dominated by actinobacteria, minimal representation of Bacteroidetes, and certain representation of gram-positive firmicutes. But unlike firmicutes present in a cecal microbiota, which mostly belong to Lachnospiraceae and Ruminococcaceae families, firmicutes represented in the litter microbiota are those belonging to family Staphylococcaceae and different species from Staphylococcaceae. Well, this is all what I wanted to share with you, to tell with you right now. If I'm to summarize what I wanted to tell and what I want you to take with you home, back home messages, definitely there are new opportunities for characterization of complex microbial populations in whatever type of samples. I mentioned cecal or intestinal samples, I mentioned litter, but you can use it for skin microbiota, you can use it for characterization of feed microbiota, whatever you want. You can use these novel technologies for characterization of whole genomes. If you purify gut anaerobes in their pure cultures, you can sequence them, predict their metabolism and so on. You can use the same techniques and same instruments also for characterization of complex microbial population. and applications are nearly unrestricted. You can do whatever you want. And if you purify RNA, a messenger RNA, and reverse transcribe this mRNA into cDNA and sequence cDNA, you can use the same platforms, same instruments also for characterization of gene expression. If you are within a microbial population, so no expression and so on, you can use, and if you are within poultry production, you can use it. Target could be chick, as I already mentioned, intestinal, respiratory tract, urogenital tract, whatever you want, skin, eye, anything. But within the poultry production, you can also analyze microbiota composition in litter, as I mentioned, dust, air, feed, water, anything. And finally, you can also use, with a certain care, you can also use sequencing as a diagnostic tool. From time to time, we are contacted by farmers with unspecific disorders in their flocks. And what we suggest them is that we can sequence. We do not promise anything real because we do not know. Sometimes it works, sometimes it doesn't work, but not directly with the poultry production, but within a human diseases. In one of the cases, permanent diarrhea and issues with diarrhea in a human being, we sequenced, and surprisingly, a little bit unexpectedly, at least considered to the conditions in the Czech Republic, we identified that this person was, its fecal microbiota contained 3% of Yersinia enterocolitica. Clear pathogen when it is set, cause of the diarrhea, cause of the intestinal problem, but less common in the Czech Republic, and therefore the pediatric medical doctor was unable to diagnose this disease initially without our assistance. So yes, that's all for the moment from me. Thank you very much for your attention, and I will be happy to take your questions.
Thank you so much, and also from my side, welcome to this webinar. This webinar that is a teaser for the real thing next year in Cartagena, where we all, of course, hope to see you in live. And also in that seminar, we will have a Q&A sessions, and you will be able to also interact with the speakers even after the sessions. But we have foreseen a couple of minutes for a Q&A session now. The questions, there are a lot of questions coming in for Ivan. Ivan, a great job. Thank you for kicking off for this session. Maybe I'll try to select a couple of questions because I don't think we'll have time to cover all of them. But maybe let's select some of them. The first one would be, does the microbiome composition that is measured depending heavily on the primers being used, does that change the outcome of the data?
Well, it's a difficult, obviously. Anything can influence the final output of your analysis. It's not only the selection of primers, but it is also the mode of DNA purification, use of Taq polymerases. The potential influences and potential issues are on a very single at each individual step of your analysis. Final computing also may dramatically affect the output. So answer is yes, primer selection may influence, but well, I'm not thinking, usually my life is not like this, right? I always say to myself and to my colleagues, here you are, there is a material and methods chapter, and in there I clearly describe that I'm using these primers, such primers, and that I strictly or rather recommend to use one primer pair across your own studies in your laboratory, and do not change from one experiment to another one with change the primers. Trust yourself, use the primers which you can test different primers at the very beginning if you are introducing the field. We amplify over the V3 and V4 variable region of 16S rRNA, and we are happy with this for the characterization of chicken gut microbiota.
Clear. Maybe just picking up on that, do you have an idea of researchers in this field working in the poultry industry? Would they, all of them, more or less use the same primer sets, or is this really depending on the researcher, or would you have some kind of a, let's say, a common panel of primers to be used, which would allow, of course, the results to be more comparable amongst each other, or is there not such a thing like a gold standard set of primers for research in the poultry industry?
Again, I can't answer really, because I'm usually looking directly in the results chapter of different studies, and I'm immediately thinking of results. I would say, but it can be biased by my personal experience. I would say that people usually use really an amplifier over V3, variable region 3 and 4 of 16S rRNA gene. But well, now trust me with, let's say, 80%. Do not trust me 100. I might be mistaken here.
Okay, all right, it's clear. Maybe next question, can we, by studying this composition of Zika, you've shown us nice overviews of data, but can we also use this data to predict, let's say, healthy chickens versus diseased chickens, or maybe even going a little bit further, can we predict performance? Can we predict what kind of compositions could be more productive than other ones? Yes, are we ready yet to make that kind of conclusions?
It is pretty difficult, and again, I do not. If anybody is saying whatever, linking really the performance, and if somebody wants to be really convincing, it's if somebody is too self-confident, I will be very careful, right? Honestly, I don't think that anybody can tell you what microbiota composition is ideal one, the most optimal, and so on. At some conferences and during some discussions, I spotted the discussion, and this is something what I would agree probably, that more important, perhaps more important than the composition itself, is the stability of the microbiota. So when the microbiota composition is stable over time, does not change that much due to different reasons, it could be changes in the feed, then this is more important than the composition itself. There are individuals which microbiota which consist of 90% of gram-positive firmicutes and 10% of Bacteroidetes. There are individuals within the same flock microbiota of which consist of exactly the opposite, 90% Bacteroidetes, 10% of firmicutes. Both of them are equally healthy and equally correct. So at the moment, I don't think that anybody really seriously can answer that yes, one particular bacterium or particular microbiota members are really gold standard. There might be positive correlations. Faecalibacterium, for example, is considered as one of the markers of a good microbiota structure. So the more Faecalibacterium, usually the better performance. But those are rather observational conclusions, correlations, and I don't think that this is a real proof.
Yeah, I maybe refer to another question from an attendee, and he was asking, was there a correlation between types of microbiota and production parameters or resilience, or the environmental outcome of food production? I think what you're suggesting is that that correlation with production is not there, but with resilience, there is a lot more a correlation with that if you have a stable microbiota. Would that be the right conclusion for the moment?
Yes, here I agree nearly in a full extent. There are commercially available products which consist of products of competitive exclusion, right? So these products consist of complex, undefined chicken microbiota. And whenever we tested this efficacy of these products for our own control purposes, yes, chicks colonized by these products are much more resistant, for example, to colonization by different enteric pathogens like Salmonella. So with a gut health status, yes, complex microbiota, adult type of microbiota indicates high resistance to infections, different disorders. When I am to really associate the microbiota composition to production parameters, we at the moment do not observe anything like this, or if then we observe something sometimes, and when we repeat the experiment, the effect is gone. So maybe sometimes in the future we will come with something even in this area, but not at the moment.
Great. Maybe a last question, not in order to go over time too much, but I thought this one was also an interesting one. What are your views on which samples we should focus on for representing the gut microbiota? Are fecal samples or cecal samples which allow longitudinal assessment, are they sufficient? Yeah? Fecal, there's already a difference there. What would be your suggestion?
Well, here it's again quite clear, but I might be longer in my answer than it is possible. So if it is possible, if there are no problems with ethics, no problems with longitudinals, no requirements for longitudinal sampling of the same chick or chicken, then I strongly advocate and recommend to really terminate the experiment, sacrifice, and collect cecal, colonic, duodenal, jejunal, ileal samples. Because you, in this case, you have everything under full control, and you know what type of intestinal tract you are sampling and what you can expect then. Whether to go, why to avoid fecal samples? Well, the reason is within physiology of chickens, right? Most of the fecal samples, fecal droppings, is a digester which passes directly from a small intestine into colon, and then it is expelled from the organism in a form of fecal material. That type of fecal material will mostly represent small intestine microbiota, because it goes directly from a small intestine directly through the colon and outside. So you will find out that it is rich in lactobacilli, which are characteristic for small intestine. Mistakenly, correctly, but mistakenly. But twice a day, usually twice a day, maybe 3 times a day, this type of fecal dropping is replaced by cecal excreta. Chickens, small amount of small intestine digester goes into cecum, is fermented for 12 hours in the cecum, and then the content of the cecum is voided and expelled as a fecal material. And if you do not keep a control, an eye over that type of samples, cecal droppings or true fecal material, you will introduce an uncontrolled error and mistake in your studies, right? So this is why if you sacrifice and perform post-mortem, you know what you are collecting. If you collect fecal material on the floor as a part of litter, you start to lose that type of control. But of course, if you have to go for longitudinal studies performed on a single individual, you have to collect fecal material and determine fecal microbiota. But my preference is really, if it is possible, then have a control over the samples. So avoid fecal.
Excellent. Thank you for the answers. We have more than 20 questions open still, so I think that reflects a little bit the interest in the topic. We will pick up on this topic, of course, again next year as well in Cartagena. But for the moment, I think I will just have to remain with thanking you for the excellent contribution and the excellent Q&A session. And if you want, you can do some homework with trying to answer all these questions that are still open. So we'll send you the list, so you can see if you have interest to answer them, because most of them are anonymous, so we can't find the question takers anyway. Now, switching to the next speaker, it's really my pleasure to introduce to you Professor Dana Stanley from Central Queensland University in Australia. She's a real leader of a microbial genomics research cluster focusing in not only poultry, but livestock intestinal health in general. And also, of course, derived from that probiotic and next generation antibiotic development and pathogen control. She's not only an excellent researcher, but she's also passionate about nature conservation, animal ethics, exercise, and nutrition. I would say that would be an interesting topic today with a title, and I'm trying to find the exact title, here with microbial metagenomics, transcriptomics, proteomics, metabolomics, tools, application, and perspectives. That seems a lot for 20 minutes, but I'm sure, Dana, that you will you try to make it a comprehensive overview in this 20 minutes. The floor is yours.
Thank you. Well, thank you for your introduction, and thanks, Philip, for inviting me, and thank you everyone for attending today. Again, thank you, Ivan, for setting up the scene for my talk, because my talk is really building up on the previous presentation. I am going to talk about microbiota analysis and analysis of gut intestinal community using other methodologies, other omics methods, in addition to 16S. But before I continue with the talk, I will just stress the major difference between microbiota analysis and microbiome, or amplicon sequencing and metagenomic sequencing analysis. So when we do 16S microbiota analysis, we extract DNA, and then we amplify one gene, 16S gene, and then sequence that. And what we get from this data set is taxonomy, as Ivan presented. You've seen phylum level, genus level, and whatnot. On the other hand, when we sequence metagenome, we sequence all of the DNA, so all genes in the community. So metagenome means all genes in a community, meta-transcriptome, all transcripts in a community, meta-proteome, all proteins in the community. So it's easy to remember what metagenome is, all genes. On the other side, what we get from metagenome is insight into microbiome, not microbiota. And microbiome has many definitions, but it includes taxonomy. It includes 16S data, type of data, but it also includes the genes. We're sequencing all genes, not just 16S. And from those genes, we can actually assemble those genes into pathways and into functions. We can look into resistome, we can look into viruses, then we can assemble metagenomes of unknown and unculturable sort of organisms. We get much more data, but it's almost 10 times more expensive. It's much more expensive. Both of these, as you can see here, will give us taxonomy, but it's different type of taxonomy. So it depends what you want from your data set. Again, of all of my presentation, what you're going to pick depends on how much money you have. But let's just imagine we have unlimited budget, which would be great. And so if we want to look into whole bacterial community, we still want to do 16S, because when we do microbiome analysis through metagenomic sequencing, and we look into taxonomy from metagenomic, not 16S data set, we will get very few bacterial species as opposed to 16S. On the other hand, there are a lot of issues with 16S, and more we use it, more apparent and obvious they becoming. Among those issues, we have issue of primer choice. So you have to choose primers, change primers a little bit, you get completely different data. That's because each primer set will take a handful from the microbial community and will pick up only one specific type, a specific group of bacteria. You change primers, you're picking up another handful. The other issue is that we also have copy numbers. So some species will have 15 copies of 16S, others will have one. So it looks like the ones that have 15 species are 15 times more abundant. It's impossible to properly account for copy number. And we also have other issues with that. So if you take all of these things into consideration, another important thing is that with microbiota, we can get genus level, okay, but we cannot pinpoint anything at the species level. For example, if we take OTU and blast it against microbial databases, we can get 10 top that have 100% identity, and software will pick the one of them. So it's not really reliable at species level, even if it tells you, even if software tells you species level, species, even strain level. So again, I want to just stress that it depends what you want from your data. One of the main issues with taxonomy, which we get from 16S, if we focus on taxonomy, is that taxonomy is extremely variable. The variability of that is very clear from this Nature paper published by Human Microbiome Project Consortium. What they did, they collected so many samples from all around the world, and the conclusion is that microbiota is, by taxonomy, even here, as you can see at phylum level, is very variable. It's variable between the people, it's variable inside the same person. If you take sample, let's just imagine that we take the samples from all of us today in this virtual conference. If we look into our taxonomy, the species we have or genera we have, it would be very difficult to find core microbiota that we all share because it's so variable. But the functions we have in the gut, molecular functions, will be very comparable. Actually, most of functions, according to this paper, are core. This kind of makes taxonomy irrelevant if we want to do intervention and change something in the treatment. Say if we have a treatment and we want to intervene into disease, targeting taxa is not going to give us much. Maybe restoring logo for the future, despite the price. Again, we can look at our data at gene level, but we can look at it at protein level, enzyme level, category, K categories, and so on. And this allows us to get a lot more information as opposed to just looking into taxonomy. So what is so when we look into this paper, and I'm sorry that I cannot get this of the presentation. Sorry. I'm sorry, I'm trying to get rid of the messages as well. So unfortunately, we can actually look at the things at enzyme level, and this manuscript, whose title you cannot see, unfortunately, is showing, it's a review paper that is showing how many new enzymes were discovered looking into culturing of uncultured bacteria. So, and you can see here that we have a range of metagenome sourced enzymes that are commercialized by BAS, for example, and many other companies. The other thing that we can get from our data is the resistome analysis. And resistome analysis comes as a bonus. The reason resistome analysis comes as a bonus is that we can just take, instead of doing just functions and gene categories and whatnot, we can take the same sequence and just blast it against resistome database and get a resistance profile for each bird, for example, if we're looking into chickens. So this is very interesting because nowadays we think that we removed antibiotics and there is no more resistance, but there is resistance in chickens, in pigs, in human, and sure that my resistome could beat the resistome of this chook. This is a resistome of layer chook that had about 150 different resistant genes. We have similar situation and we look into human data. So resistance is a natural phenomenon, but interesting thing that came as a side effect of looking into this type of data, which I just started looking at because I can, not because I targeted, is that when we look into some nutritional treatments that target, for example, nutritional treatment like short-chain fatty acids or other acids or even mineral supplements, range of these treatments actually can significantly reduce and control resistance, and this can be actually used to try to control and reduce resistance even more than just removing antibiotics. We can also look into viruses, and that is another part of this type of analysis. Now, if you are thinking, I would so much like to get rid of this. Oh. And I cannot because I can't do it on a laptop. I'm so sorry. Okay. So if you now thinking, all right, I just learned how to use Chime, and I just learned how to, and I just can finally afford to do microbiota analysis, and you're telling me now that function is the way to go, do not despair. There is PyCrust, and there are other methods that can be used to actually to infer, to predict function from the metagenome, from the 16S data, from sequencing just 16S. And PyCrust as a software has been validated in many different diverse communities, and it's got about 85.33 according to the paper correlation with metagenomic data, and it will give you functional predictions, not functions based on gene sequencing, but predictions. And it still is very good way to see if it's worth paying the money to go and sequence actually metagenomic data. So again, we want to move now into metatranscriptomics. And metatranscriptomics is answering multiple questions, different type of questions. So with 16S, we're answering who are they? So what different taxonomy we have in the community. When we look into metagenomics, we look at what genes do they have, meaning what is genetic potential, what could they do? But when we look into metatranscriptome, we're looking into which genes are expressed, which means what are they doing right now, how they're responding to treatment rather than what genes they have. In this case, we sequence total message of the bacterial community, message RNA. Now, metagenomics shows us one type of view, and it's very precise unless we want to specifically look what one treatment triggers in that community, and then we need to do message. Then we need to do metatranscriptomics. But message does not have poly A tail. That is now opening lots of problems for if we want to do this. For example, when we do metagenomics, we will pick up taxonomy that is not just 16S based on one primus. We'll pick up helminths, we'll pick up eukaryotics, we'll pick up amoeba, whatever is in there. When we talk about looking into message of the whole community, we have to pick. So based on the kit we want to use, we have to, we cannot pick them by poly A tail as we would for eukaryotics. So bacterial has different kits for purification and so on. It gets lots of ribosomal RNA contamination, so a lot of listed sequences. Kits are getting better every day. But message RNA from microbial community is less stable than message from, say, chicken liver. So we not only that we need different kits for eukaryotic and prokaryotic, we need different storage. The people usually suggest extract RNA immediately. Don't even freeze it. Do not ever think of using RNA later because it will not work with bacterial or microbial community. Again, we have issue with databases, but databases are not enough bacterial genomes and whatnot, but they're growing really rapidly. And again, we have some reference issues and whatnot, but we do get lots of interesting data if we look into this. On the other hand, if you compare metatranscriptomics with metagenomics gives you the other extra things, as you remember. So you can get resistome, you can get other things as well. So if you have money, ideally do both, and both is better. More is merrier. So now I am moving into proteomics, and metaproteomics is a step from genotype, looking at genes or transcripts, to phenotype, how these genes are expressed and what are the products of these genes. This is not sequencing based. So most of the stuff I presented so far, and that even presented as well, you can do all of it on HiSeq, on OverSeq, on a very similar methodology. Now we're moving into analytical chemistry lab in metaproteomics and metabolomics. And that's completely different story. We have a range of instruments that are very different to each other, and it is fundamentally different, but that can do the same thing different way. Lots of genotypes do not correspond to phenotype. This is the logic behind doing metaproteomics, which means that, for example, yes, we do have message and we know what is being transcribed, but not every message will end up in protein. But is that enough to justify $800 a sample? It's up to you. the literature agrees that the major significance of this methodology is if you have biomass differences, because other methods are focusing on numbers of cells and are based on number of cells, but proteins will tell you biomass. So if you have much better cells, you get much more protein, even if the cell number is the same. So that's basically where the advantage is. Very useful methodology, but still quite expensive. There are two ways to look into proteomics. There is bottom-up and top-down approach. Basically, bottom-up approach is working on cleaved proteins. So you take trypsin and you break the protein in little peptides, and then you sequence them using these instruments, and then you assemble those little peptides back into protein again, reconstructed from the peptides. This is also called shotgun proteomics, and shotgun, whatever it's called, shotgun means you're looking at everything at once. Basically, this is happening when you have many complex unities and you want to see the profiles. So what is the difference in profile? You're going to see the heat map, PCA plots. Are they similar based on everything that's in there or not? Top-down proteomics is looking into identifying and quantifying even unique proteoforms. So much more precise, but is not as wide. So you will focus on specific proteins for which you will have standards. And so you will take your pick. You can do it up to hundreds of proteins using top-down approach, but rather than thousands using shotgun. But what you're going to get is you can nail it up to a picomole or nanomole. So you can nail the concentration and you can track proteoforms, different forms. You can look at sequence variants. You can even look at post-translational modifications, but you will never discover anything new using this approach because you actually have standards and you know what you're looking for. But if you shotgun, you can actually see some proteins that are different between communities and then focus on them going down and using top-down approach to try to actually look at them a bit more precisely. Now, metabolomics is the closest we can get to type. Metabolomics is actual end of the story. If we look into here, genome is genome. Genome is not going to change no matter what you hit it with. Genome is very stable. Transcripts are responding to treatments. Proteomics also will be responding in a similar way to transcripts. Proteins will respond. But metabolomics or metabolites will be responding to treatment the most. They will be changing the most for example, if you take a lolly and put it in your mouth, instantly you can feel the sweet taste. And that sweet taste is produced by these metabolites that have already traveled to your brain and sent the signal that we're talking about something sweet. Obviously, metabolites are products of enzymatic reactions. And we can also look into host metabolomics and we can look into microbial metabolomics. So we can look at both. It's becoming very popular. We have precision metabolomics, personalized metabolomics, because suddenly we have the instruments that can allow us to look deep into this area. It's an unexplored area. You can discover a lot of things. Mammals have a lot of metabolites, but microbes probably double or triple that, and plants maybe even up to 10 times that. Plants have a lot of metabolites. The main advantage is that 95% of diagnostic tools and about 90% of therapeutic tools are actually small molecules. So this is because we can really give accurate concentrations. If I measure butyrate here in Australia and someone measures butyrate in Belgium, and we have the molarity, the data should be really comparable. And we cannot really achieve that level if we look into proteomics, for example, for diagnostics or even therapeutics. So it's great for biomarker discovery. Again, we have shotgun and targeted and untargeted methods. So targeted, let's say I just want to see short chain fatty acids and I tell GCMS, just look here in this range where I got butyrate and in this range where I have acetate and so on. Untargeted is when I want to see every signal that is metabolite in my data set. And if I do that untargeted, I can get up to 30,000 features. But the problem with method is at the moment, we can probably identify maybe 1, 2, 3, 5% of the features that we have. Other features will be predicted by instrument. MS, mass spectrum will predict name and structure of it, but we probably won't know what it is. Estimated number of chemicals that are actually building blocks is in millions, whereas when we do sequencing, we have ATCGs, we don't have much. When we do proteomics, we've got 20 amino acids, but here we have much, much more diversity. But again, we have issues like different extraction methods. For each different type of metabolites, different extraction methods, different for lipids, different for carbs, different proteins. So we kind of actually narrowing down these methodologies. So in a way, proteomics is also a type of metabolomics, depending on the size of the protein. So again, when we look into these data sets to put it into pathways, to look into categories like functions, microbial pathways that are currently in KEGG are based on human annotations. So microbial community has so much more, and we just getting the data from the top, and it's a bit difficult when we do shotgun sort of approach, but targeted approach is mostly used. And again, different methods will give you different number of metabolites, different sort of concentration range. One example of this is of usefulness of these methodologies in this paper. And this paper is it's a review paper, reviewing quantum sense things we got from looking into bacterial metabolites. And what this tells you is that they're not only communicating with their own species, but they are capable of listening and broadcasting images and signal to unrelated species. They're even capable of changing and altering each other's behavior, and also signal and communicate even with little multicellular eukaryotic organisms. This means that microbiota speaks metabolic language. So metabolites are the language of gut microbiota, and LCMS is like universal translator, but it will take us many, many years to translate what they're saying to each other. Again, we also want to look into host interaction with microbiota. Often we do look into RNA-seq, maybe of ileum or cecum, and we'll get list of genes. And a lot of classic analysis, people will say, oh, these genes are up, these genes are down. And a lot of these genes that are up are in this category, therefore this category is up, which is wrong way of analyzing the data and brings wrong conclusions. But what we can do, actually, there are currently softwares that we can use that will allow us to look into upstream analysis and do disease and toxicology prediction model just doing RNA of the host, and then link that to all of these omics methods that I just described. Here is an example of disease heat map generated in IPA. The top diagram, red means activation of disease, blue means inhibition of disease. Top heat map is E. coli infection, systemic E. coli infection, and this here at the bottom blue heat map is chickens fed olive oil poultice. So whatever is left after you take oil, of olives. And this is the end, I promise. So I don't have time to go through all of these issues that we faced in all of these methods. I could talk about them for quite a while. I'm going to mention just power analysis. And this is because if I do post hoc analysis of, say, RNA-seq, and I know what variability I have from actual data, and I recalculate the power, it will tell me I need 60 replicates, for example. That means about $50,000 to do one experiment. Well, no one does that. Does that mean we are underpowered? Because when we actually do qPCR confirmation, we can confirm all the genes pretty much exactly as we get them if we do 5 replicates instead of 50. Again, there are a few other issues with these areas, but at the end, my final slide is if we looking into gut microbiota, we have to look into hosts. So everything we do in everything I presented to do for gut microbiota, we should ideally do in the host. And if we had millions of dollars to spend on sequencing, I swear I could spend every last cent, because this is a fascinating area, and analyzing this type of data and trying to link it together is really interesting. It's better than playing games on the computer, I promise. This is my team at CQU. So this is Yadav, Darwin, Zipeng, Advit, Maria, Sung, and Fred. And I'm saying hi to them if they're watching. And thank you for tuning in and listening to my talk.
Thank you. Great presentation, Dana. As typically for this kind of subjects, I think your presentation raised more questions than it answered questions. So, but I think that was a little bit the purpose. Let me try to select a couple of questions that popped up. Maybe the first one, rhizostome identification is a great tool, but does that mean that it might be that you detect genes here that are not leading to phenotypical resistances? Would you have some insight?
Yes, so that depends on, there are lots of different rhizostome databases, and these databases contain actually confirmed and proven genes. That the fact that the birds are carrying these genes is the purpose of doing the rhizostome. So they are carrying those resistant genes, they can exchange them, and we can actually use actual metagenomic sequencing to actually pinpoint and see which bacteria are carrying which resistance genes to see whether pathogens are actually, or which specific resistant genes are carried by, for example, Salmonella. We can actually do that. We can do it by species, and it's much more information on what is going on. The situation with resistance is, yes, it is reduced, but humans, I would say, have the most. It's not just blaming chickens or something. It is natural phenomenon. If we take 10,000-year-old mummy, Egyptian mummies, when they extracted DNA from the gut, had all resistant genes to all known antibiotics, even the ones we just discovered, which means it was there and it will always be there. But if we want to try to reduce it, look into rhizostome now, using this technology, and looking into, it's very, like ascorbic acid is going to make bacteria lose their plasmids, and is a COIN, which means conjugation inhibitor. And that means that simple things like ascorbic acid and a lot of other, actually a lot of fatty acids can make bacteria lose plasmids and prevent spread of antimicrobial resistance. And it's an area in poultry nutrition that would be interesting to have a look at.
Great, great. Maybe another question. Do we know which functional microbial pathways are really crucial for gut health? The really crucial ones? Would you have an idea yet, or is that a little bit too early?
So you can look at them at different levels, just like you can look at phylum and then get all the way down to species. But at the top level, the main sort of functions for human, for example, have been defined in human microbiome projects, and and you can actually pin them down all the way to the sort of deeper levels. Obviously, those are metabolic functions will be always there, and there is a range of microbiota functions. Interesting thing though is that when we look into treatments, for example, we do analysis of gut microbiota and functions they have, and then we look into functions of ileum of chicken, and say we add the treatment. Treatments are going to do the same thing to the host functions as they will do to microbiota functions. So it is interesting to look where this functional analysis will take us, but definitely core functions for functioning of gut have been defined by Human Microbiome Project.
Mm-hmm. Okay, so maybe as a teaser, maybe also would you would you from there, would you see on this moment, like, let's say, massively applied omics in human health or in nutrition or animal health? Do you see any omic technology being broadly used already with results that yield better health or better economics, or is all this still in a more experimental, finding, exploring phase? Can you help us with that?
So unfortunately, I think that omics will remain a privilege of well-funded research teams. Although 16S is reducing in price, and everyone thinks that sequencing prices will go down. Well, sequencing is not a problem anymore. Sequencing is very cheap now. It's the kits. So library preparation kits are still in thousands, and if you have and they are, I don't know, cover some X many samples, and if you have a lot of samples and then you add the price of sequencing. So I do not expect that sequencing is just going to keep plummeting down as many people think. At the moment, I think the sequencing prices are going to maybe go down very slowly. And if, as I said, if we had unlimited budget, we could dive into multi-omics, but unfortunately, unlimited budgets are fantasy that of scientists like us. So it is going to grow. In my opinion, people will start doing more and more. I cannot see that proteomics is going to, my last quote for proteomics was $800 a sample. We have instruments at CTU, we have LCMS, MS, triple quad, and we can do targeted proteomics. We cannot do shotgun. We do not have MALDI-TOF or whatever instruments that are needed for that. But we still staying with ELISA for that because really doing everything would just be an overkill. And my choice is to do 16S microbiota and then metagenomics for functional analysis. And I always do host RNA-seq. And I try to actually target specific proteins using other methodologies, but we do want to focus strongly on metabolomics because I think metabolomics is not as expensive as other methods, and it's the future. It will grow very fast, I believe, in next few years.
All right, that's a clear answer. Thanks for your fantastic input. And then I would like to switch to the next speaker and thank Dana again. Usually there's a huge applause now coming from the crowds, but unfortunately now we don't have that. We should have some kind of a virtual clapping tool or something, but we don't have that here, Dana. But anyway, thanks from the organizers' side. Then it's my pleasure, my honor to introduce to you the last speaker of today, Professor Ducatel, who's also my personal professor of pathology when I was graduating as veterinarian here in the University of Ghent. And Richard's current research is mainly in gastrointestinal health with a focus on poultry, cows and pigs. He's mentoring over 30 PhD theses in this field of research in the last couple of years. Professor Ducatel has been authoring or co-authoring over 695 scientific publications and more than 400 abstracts. He has been a keynote speaker at over global congresses. I don't have to express even more how important Professor Ducatel has been and still is and will be in the field of poultry research. And today, his topic will be, let's say, an integration of what the previous speakers have been talking about. He will drive us into the field application of sequencing and omics data, and he will do that from a veterinary perspective. Professor Ducatel, the floor is yours.
Thank you, Martin. You made me feel very. You made me feel very old, but anyway, when I was given this task to look at the applications as they are today in a veterinary perspective, I realized that I should sit back and just ask myself the question, in veterinary medicine, where do we come from, and where are we going to? And of course, you know, in the old days, everything was probably much more straightforward. We had cost postulates, and when there was an infectious agent and it could cause disease or even mortality, you could start thinking about developing a treatment or a prophylactic measures, a vaccine. But since then, of course, we've moved on, and we've been facing other types of diseases, which we call multifactorial diseases, like, for instance, the respiratory syndrome, which is known in many different animal species, which makes things more complicated. And even more so, today we have these things which we hardly can call diseases, defined diseases anymore, but we call them complex health issues, as there is dysbiosis. And of course, in this complex context, that's where omics technologies come in the spotlight. And of course, you know, as with each new technology coming available or appearing in the veterinary field and in the field of animal production, you know, at first it's the scientists that are interested and that are fascinated and that are becoming sometimes over enthusiastic. And I was very glad to hear from both Ivan and Dana that we need to be sometimes a little bit critical at some points, but what we definitely should do is we should go for it. And even if at first it's always rather like, right, I've been able to do and to work with this technique, and what comes out is at first it's very descriptive, and it still is at this point to a large extent. It's descriptive. It's just reporting under these conditions we see these changes in the metagenome or in the metatranscriptome or whatever. But anyway, I strongly believe that the positive thing about omics is that it is showing you the broad picture. It's like a more holistic approach to complex situations, which may be complex situations in the healthy animal, like, for instance, the intestinal microbiome, or it may be complex situations with respect to disease, like, for instance, when the microbiome is shifting away, and we call it dysbiosis. In the past, we were all looking at things from our different point of view, and we were looking at the specific aspect, and it made life very confusing. And I believe that the omics technology will bring scientists more together and will give us a more complete view of complete picture. Oh, and by the way, that was me, of course, always at the wrong end of things. But what we get now is a situation in which we have, even if it's a blurred picture of the entire issue which we are focusing on, now we can say have a more scientifically sound decision on which detail we want to focus on. If we want to focus on the skin of the elephant, then we'll know why we want to focus on the skin. And I want to give you one typical example where we have used 16S sequencing, not more than that, and to focus our further research. And that was when we were looking at a simple feeding trial where we compared a control group with a group where 0.5% of cellulose was put on top of the feed formula in the starter and 1% in the grower phase, and we were doing a analysis of the performance of the animals, and we were astonished to see that the birds that had the cellulose in their feed had a better feed conversion than the control group, which of course was against the traditional belief that cellulose is a filler, especially in the newborn, the newly hatched chick, and even and that reflected in the feed conversion and in the daily weight gain. And when we did the 16S sequencing and we looked at the microbiota composition, I I will spare you the details, but it turned out that there was hardly any difference except in the phylum of the Bacteroidetes, which Ivan has already referred to as an important phylum in the adult birds, but in this case it was the young chicks, and within these Bacteroidetes there was a difference only in the family Rickenella, and specifically so in the genus Alistipes, which appeared a couple of times in Ivan's presentation already. So we were fascinated by this very specific response of the microbiota to this specific feed supplement, which we gave to an otherwise harmless and very standardized commercial feed formula. And so it turns out indeed that Alistipes is part of the adult chicken microbiome, but it's not only in the chicken Zika. When you go through the literature, it's a dominant genus in the cecum of healthy rabbits, and also in the colon and cecum of the Asian elephant. And so these are typically fiber consuming and animal species that live on a lot of fiber. So that sort of confirmed us that this must be something key and something very important with respect to this cellulose. And of course, the other people from Australia, the first author of that paper a long time ago already was Torok, and they were comparing, the microbiota of well-performing broiler flocks with poor-performing broiler flocks, and it turned out that Alistipes was associated with better performance. So that was again reassuring us that there was something important about this Alistipes, and it has been documented indeed that Alistipes, its main end product of metabolism is succinate. We should realize, of course, that succinate can be taken up by the epithelial cell because there is a specific receptor for succinate on the intestinal epithelial cells. So it's taken up by the epithelial cells. And so we thought that, that's clear. Okay, succinate can be used in the Krebs cycle and can be consumed by the epithelial cells, and it will provide energy to the intestinal epithelial cells. Not so. It turned out that there was a paper coming out already in 2016 in mouse, knockout mice models, in which it was very elegantly shown that when succinate is, I will just put my pointer on, when succinate is produced by the microbes in the colon of the mouse, it is taken, there is receptor-mediated uptake, that's correct. But then in the epithelial cell, the succinate is converted into glucose, and this glucose is passed into the bloodstream. And so this is an important aspect of intestinal gluconeogenesis, or in other words, the day-old chick can cover episodes of darkness when it's not eating, avoiding hypoglycemia by extracting glucose indirectly from cellulose. And we confirmed this in vitro by showing indeed that Alistipes can convert cellulose into succinate. It is the epithelial cell that is responsible for the gluconeogenesis. So that explained to us why these chicks were doing better, simply because they had this episode of darkness which they could overcome due to this cellulose being present in the Zika. And that, of course, is becoming very important because we know that hypoglycemia is a major cause of sudden mortality in broilers. And it turned out that since we published that paper, it has been realized also that in humans, Alistipes is there to degrade cellulose as well. And we know that human beings are on average able to break down and to metabolize about 1/3 of the cellulose in the food And that is, of course, a considerable amount of glucose that can be produced in this way. So that was just an example of how omics can steer your further research when you dive into a certain aspect of a certain scientific question. But of course, the main topic for me was to what can veterinarians or even or farmers do with omics, and as Dana already told you, we should not be over enthusiastic, simply because if what a veterinarian wants is immediate results. He wants to take a sample and he wants to send it to the lab, and the latest he wants the results back is tomorrow, preferably yesterday. So that's not possible, simply because it's still for a lot of these tools and a lot of these techniques, data analysis is the time-consuming, effortful task that needs to be done by experts. So that is a limiting factor. And the other thing is interpretation of data, and Martin already alluded to this in the questions he put forward. He insisted on putting these questions, and quite right actually, because you can get these huge amounts of information about different metabolites being produced and different genera being upregulated or down, or becoming more enriched or reduced in richness. But what does it all mean? And so what the veterinarian wants is straightforward interpretation of data, and preferably immediate practical conclusions. Now, what the veterinarian doesn't really care about too much is maximum accuracy. We can live with a little bit of it doesn't need to be perfect, as long as it helps in our day-to-day job of keeping the birds healthy. And of course, it should be economical, and Dana already alluded to this, that the cost can be at the moment still can be restrictive. So if sequencing is not really possible today, maybe we could sort of take one step back and boil things back to something which is affordable and is faster, and that could be if you could select the right probes for, say, key microorganisms in the intestinal tract, and you can put these on a chip, as is the case, for instance, in low density microarrays. They are affordable, and you get about 200 probes. So if you select the right probes, you could get albeit a blurred picture of some important key organisms in the intestinal tract. And so there are, at this very moment, there are companies offering this to their customers as a service, as a surrogate to the sequencing and to the more sophisticated technologies. But of course, it is, I think, reasonable to believe that sequencing will come closer and will become like something that is almost like what we call pen-side, and that moment is much closer than we think, because if we go to the sequencing technologies, we know that of course sequencing by synthesis is the reference today. It's Illumina. And that's what Dana was mostly referring to, I presume. But there are other sequencing platforms that may be more economical, and one of them is for instance the nanopore sequencing. The advantage of nanopore sequencing is that it produces long reads, so it seems like have an advantage as opposed to Illumina, which produces short reads, which is responsible for the fact that you cannot go down below the genus level in principle. With nanopore, in theory, you could. The only problem is that with nanopore, you get about 2% errors, and that's huge in terms of sequencing results. So there is the principle of nanopore sequencing is relatively simple. If you have the double-stranded DNA, it is unwound, and the single strand is passing through a pore in a membrane, and as the different nucleotides pass through the pore, there are different ions coming with it, and this what they call disruption ion current across the membrane that is measured in this system. So it's a very straightforward and very directed way of sequencing, and the machine that is used for that is not an expensive Illumina sequencer, but it's this simple device of €1,000 in which you put these flow cells of between 450 and €800, which can be recycled, so you can use them multiple times, and then you get the sequence. So that seems like something that could be useful in veterinary medicine. And indeed, there is a spin-off company from Ghent University that developed a swab for sampling, and with this special swab, and it's it's like a simple device that is used for sampling. It's a swab and a filter system, and the idea is that you take a swab from a biological specimen from an animal which you suspect of being ill, any whatever viral or bacterial disease. Then you're expecting that there's quite a lot of these virus or this bacteria in the tissues. And so with this sampling kit, you get rid of the host cells, and what is left is extracellular bacteria or viruses. And that is then put through the nanopore sequencer, and the analysis of data is relatively straightforward. There is one step built in here where you add a reverse transcriptase so that you pick up both RNA viruses and DNA viruses, and in this way you can get information about unknown viruses. And so this can be very helpful, I think, and it can be a new tool in the veterinary field. So it can be applied to diagnostic cases, and I will give an example of that. It can be applied for monitoring herd health, and it can be applied to follow up epidemics, as long as the sampling is, of good quality, because you need intact host cells, intact bacteria, and intact viruses. And so, in one case, we use this technology in the diagnostic challenge which we were facing. That was quite a bit of mortality in a number of broiler flocks, which presented clubbed down and pasted vents and uneven growth. And so, other than uneven growth, there was clubbed down indeed in about 15% of the birds. 10% of the birds also had tremor, and they had these pasted vents. And on histology, they had pancreatic atrophy. And this started off within 1 week post-hatch, so very early. And so we also knew that there were issues with the avian encephalomyelitis virus vaccination in the parent flock. So this could have been, it's quite a confusing picture, actually, going in different directions. And it was quite serious because in these broiler flocks, which all came from the same parent flock, there was 30% total losses. And so we were wondering, could this be an avian encephalomyelitis outbreak? Or when you think of the pancreas atrophy, it could as well have been a paramyxovirus outbreak. We were thinking of reovirus or even an unknown new virus. So, and alternatively, if it's not infectious, it could have been a nutritional deficiency in the parent flock. And so we came to the conclusion that using the pathosense technology that there was no viruses. So it turned out indeed to be a nutritional deficiency in the parent flock. So that really helped. So I think what I wanted to say is that as we speak today, the task of the veterinarian is no longer limited to simply treating diseased animals. And what is expected from the vet of today is that he should keep the animals healthy at all times and under all circumstances. And that is a much more challenging task. We should, I think we can realize now that these omics technologies, they have already contributed enormously to our understanding of complex health issues. And there is still a lot more to come out of the scientific investigations, which excellent scientists like Dana and Ivan are doing on these topics. And of course, I strongly believe that it has its place also in the toolbox of the veterinarian. And of course, it is available as we speak today. It is available for diagnostic purposes with respect to infectious agents, even if there are some still a number of limitations, like for instance with respect to the sampling, that is still very, very critical. But I think we should not forget that in the end, it's the veterinarian's responsibility. In my opinion, omics is another tool in the toolbox. Like we have added new tools in the toolbox roughly once every 10 years. It was in the past there was ELISA coming up, there was immunohistochemistry, immunofluorescence coming up like years ago. Then we had PCR coming. Now we have the omics coming, and I think there will be. It's not just one tool. This will be a range of tools in our toolbox. But we should not forget, of course, that in the end, omics is very nice. Computer-assisted technologies are very nice, but it's still, in the end, it's the responsibility of the people to be sure that everything is running fine. Thank you very much.
Thank you, Rick, for the excellent presentation. I hope you can hear me well.
Yes, sure.
Okay, that's great. So it's a very fascinating presentation for sure. Until now, we didn't really have many questions for this, I think, because people were listening very well to you. But I had some questions that were sent to me through the WhatsApp page here. And one of the questions is I like very much. It's a veterinarian's question that asks whether the future job of a veterinarian should also involve like data analytical capacities, because should he become a data analyst somehow? And a second question that is a bit, let's say, related to this one is how long you think it will take until all these technologies you just have been explaining will play a real role in the daily diagnostic work of a veterinarian?
Yes, these are indeed excellent and challenging questions. Whether the veterinarian should become a data analyst, and not in my opinion, not to the extent that he should really dive into the analytical technologies, which are, they require enormous efforts to really be on top of to master these huge amounts of computer data that become available. That I don't think that's the task of the veterinarian. That will always be the task of the scientists and the people that are really, really skilled. So there is this phase in between where it should be, the data should become available as digestible data between brackets, which can be managed by clinicians and by people working in the field. And I personally believe that this will also include feed formulators, so that they can follow up. And I think in feed formulation, it's pretty close to being applied in practice as well. I know of some big feed companies that do huge efforts and huge investments to be on top of this technology, and because it can help you to follow the consequences of changes in your feed formula, or changes in the technology of feed production. But in the veterinary field, my strong belief is that veterinarians are now more and more involved as being responsible for the follow-up of big integrations, and so they will not be the ones that will find out which microbe is involved in which change in the feed formula, or in which change in the environment, or associated with which pathogen that has been arriving on the farm. But the interpretation of, and the translation to the daily clinical routine will be the task of the veterinarian. So I hope that as many veterinarians as possible are following this kind of meetings, even if it seems like still a bit very high-reaching. But it is today it's very close to being applied in practice, and so the data analyst is not the veterinarian, I think, but the person who will decide on what should be done based on these data, that should be the veterinarian, yes.
Yeah, okay. Well, there's more or less a similar question, and again, it's about the role of a veterinarian. There's a question coming up in the Q&A that asks how it should be with the nutritional knowledge of veterinarians in the future. Should that be much more in the education part, or how do you think about this?
Yes, I'm very glad that this question is being asked, because I'm personally very strongly convinced that that nutrition should be a major part of veterinary training, because up until very recently there was still this gap between the feed formulation and the feed companies on the one side, and then the veterinarians facing the situation on the farm on the other side. These people should sit together at the same table, but if you want to sit together at the same table, you need to be able to speak the same language. And for that, I think it's important that the veterinarians need to be very well informed about anything to do with feed formulation, just as well as they need to be very well informed about anything to do about with management and with ventilation and any aspects of that. Because that's the only way how you can tackle multifactorial diseases and even complex health issues. Because if you're facing a condition like wet litter, for instance, it's a veterinarian that will be asked first about the wet litter, and after that, if that doesn't work, then the farmer will go to the feed mill, because he will tell the people from the feed mill, hey, there's something wrong with the feed, because my barn is completely wet. But it's the interaction between the wet litter is actually nothing else but an inflammation in the intestinal tract, which is associated with damage to the tight junctions, and this damage of the tight junctions leads to leakage of host body fluids and host proteins into the intestinal lumen, and these host proteins then shift the microbiome because of this rich nutrient source that is available. And on top of that, there's the shift, there's the Warburg effect on the epithelial cells that causes the oxygen to flow into the intestinal lumen. So these physiological or physiopathological and functional issues are all interrelated and are linked to feed formulation and to environmental and management issues. Yes.
Okay, so maybe two final questions. One of the question is about the pathosense tool. I also got some other related questions in the WhatsApp group here. Question is whether we talked a lot about bacteria, and this pathosense tool, for example, is it also the case, or can it be used for viruses, bacteria, parasites, for anything, or is it just only for viruses that it's applicable?
At the moment, it's the sampling kit. The sampling kit separates larger debris and host cells from smaller particles, which are down, the level of the bacteria and down. So it's bacteria and viruses essentially. And we should realize, of course, that with nanopore, you do have a sequence. You do have sequencing errors. So it is mostly applicable to clinical infectious diseases where there is clearly dominance of a given pathogen. So there are definitely restrictions, but in my opinion, the positive thing about this, of course, that because of the software that is linked to it, you can be facing a pathogen which is unconventional and which you are not expecting, and still you will be able to pick it up. But of course, you still need to be, as a veterinarian, need to be, aware of the interpretation of the data. Like for instance, if you take an intestinal sample, and it turns out that there is astrovirus in there, the question whether this astrovirus can be responsible for a serious clinical disease is answered quite rapidly, no. So that will not be the cause, but that is the interpretation of the data, which is the responsibility of the veterinarian, of course.
Okay, so one final question, and maybe, well, one of the three speakers can reply to it. I don't think it's more or less a remark for all of you. So one of the remarks that was given previously in one of the Q&A sessions of Ivan, and also some questions in the WhatsApp group are coming on. I mean, we talked a lot about the omics data, and especially me as a scientist, I am very well aware of what we can do with it and how much it has, let's say, contributed to knowledge on many different, well, diseases, on pathogenesis of diseases, on health in general. So it's a lot of information that's coming out, and it's shown to everyone. But how can we, let's say, well, cause these data to really be used with a lot of confidence by the people that need to use them? In other words, are there some examples already somewhere available in human medicine or somewhere else that are applicable and really used a lot, that can, well, bring some confidence using these technologies to the field? So that's a general question that came up a few times. Can you give some examples? Dana, you can.
Yes, thank you. First, I want to apologize for erratic talk, because I kept getting pop-up messages all the time on my screen. It was driving me nuts, but my students told me that they couldn't see it. So regarding this question, I download everything published in Chicken and Pigs from SRA database. And talking about data sharing and getting these example data to others, there are good databases. There is one thing that drives me nuts, and that is that a lot of mega sets, even some that are published in Nature that have hundreds of amazing data, have fake annotations or don't have annotations, or they call it treatment 1, treatment 2, treatment 3, just to prevent you from analyzing it. And there is lots of rubbish data online as well in these good databases. So when you actually filter all the junk, you end up with, metagenomics with like 5 million sequences and stuff like that. But if you understand the methodology, you can actually handpick a lot of amazing chicken data sets from any of these omics methods, download them, play with them, learn the methodology and analysis, and analyze them in different, from different perspective, using, because you will get, I promise you will get much more than what fits in paper publication that people had to sort of jammed everything. And so for people that And I've had a lot of texts and emails and LinkedIn messages after the talk where people are asking, how do we learn this? We don't have money for this. And I think the answer is in actually downloading existing data sets and slowly following the every software and algorithm will have step-by-step instructions, and play with sample data and learn to explore it while you're waiting for the moment when you can afford to do more.
Okay, thank you. I don't know whether Rick or Ivan has some extra thing to come to add here about the practicability of the applications.
Probably no. I would let it up to Dana. I agree. Yes.
Okay, Richard, you have final remarks?
I fully agree.
Okay, great. So okay, so I can go to the final slide now. Yeah. So after this Q&A, so just to wrap up, I think we had a great meeting. I already saw a lot of emails and messages in the chat of people that said it was a good meeting, and I completely agree with that. We got a lot of information, and especially also learned again how well what we are doing for the moment is really collecting a lot of data that we at a certain stage, and I don't think the time is very far from here now, that we will use this in some systems that are very applicable in the field to diagnose health, gut health, performance, and to really contribute here to evaluation of compounds, of feed additives, and to monitor health in general. So we thank you very much, Martin here and me and all the organizers, and we hope to see you next year on the International Conference on Poultry Intestinal Health in April. It's organized in Cartagena, and if you look at these beautiful pictures, you should be there. And there's also a very nice program that you can find on the website, of which the link is presented here. So I thank you very much. And Dana, I'm very sorry because it's very late for you in the evening, so I hope you have a very good rest. So thank you all, and see you next year.